Welcome everyone to tonight's NPA COVID webinar on COVID vaccinations and vaccine hesitancy. Very grateful to you all for joining us this evening. Just a little bit of housekeeping. This is a Zoom webinar, so you will be automatically on mute, and for you to participate and ask us any questions, please use the Q&A at the bottom of the screen if you're using a desktop to put in your questions throughout the event. We've got a great lineup of speakers this evening, and we'd really like you to ask as many questions as you can so that we can get as much out of the event as possible. If there are any questions that we can't answer tonight, we will do our best to get back to you. So this idea for this webinar sort of came from a meeting that a few of us had a few weeks back now with Ministers Kemi Badenoch, Nadhim Zahawi, who's the vaccine minister, as you all may be aware, and Joe Churchill. And it was a roundtable that the NPA had organised, talking about COVID vaccinations, pharmacy's role, and in particular about the BAME populations that were showing hesitancy to having the vaccination. And it was really interesting to hear from Kemi Badenoch, the Equalities Minister, about how BAME has disproportionately affected, COVID has disproportionately affected people from BAME backgrounds, and yet those people are the most hesitant to have that vaccine. And obviously, the Vaccines Minister, Nadhim Zahawi, was really, really keen to make sure the vaccine rollout program went forward and met all the targets that they were hoping to, and so far we've been doing pretty well across England, haven't we? And I think there was a really powerful message that actually Ade Williams shared with us on that call, and it's that people may not trust a vaccine, but they will trust a pharmacist that either gives it to them or talks to them about it. And I think that's such an important message, isn't it? Because as we all know, as pharmacists working on the shop floors in our pharmacies, people do trust us, and actually right now, if you think about it, we're probably the only healthcare professional that is really accessible. Our doors are open. You don't need an appointment to see us. You can just come in and have a chat with any of us. And, you know, we're on your local high streets, we're in your local Pareto shops, we're in your supermarkets, we're next to doctor surgeries, we're everywhere. And so a really, actually, really crucial role that we have in supporting, you know, moving from vaccine hesitancy to what I call vaccine urgency, and really getting people to, you know, go and queue up to have their vaccines. But I also recognise as pharmacists, we're all very, very busy people, and obviously people have been asking us lots of questions, and sometimes we don't always have the time to read all the latest information coming out about all the different vaccines, their safety, efficacy profiles, and all the information, and it can all be very overwhelming for us, can't we? So what we've tried to do tonight is bring in the big guns and the experts, basically. So I'm very, very pleased to say that we've got Dr. Shirley Hopper from the MHRA, who'll be talking to us about the vaccinations and the sort of process they went through to regulate and authorise these vaccines to market. And we've got Professor Jeremy Brown from the JCVI, will be also talking to us about how the cohorts were decided and information about that to really support us when we're having these conversations with patients. I also think as pharmacists, you know, we're very used to having conversations, aren't we, about, you know, whether people should take medicines or not, the benefits versus risk. Actually, one thing I'm very, very kind of passionate about is making sure these conversations we have are person-centered. So it's not our job to convince people to have the vaccine. It's our job to answer their questions, answer any concerns they may have, and really, you know, allow them to make an informed choice about whether they want to have this vaccine or not. So hopefully this webinar will allow you all to, you know, create that knowledge base so that you can have those informed discussions with the patients that are coming into our pharmacies. And I think actually now more than ever, as we move on to those younger cohorts that are coming up for vaccinations, I think there's probably going to be more of an issue, isn't it, to have these conversations. I know most of my 70-plus-year-olds have been absolutely delighted to get the vaccine, and they've been coming in and ringing me saying, Rena, I've had it, I feel great. Yet some of my younger patients coming in, especially some of the carers, people who work in care homes, have sort of been a little bit hesitant. And I know when I was giving the flu jab to a lot of these people, they were asking about the COVID vaccination and, Rena, will you have it? Won't you have it? What are your thoughts on it? So I think actually the time is really, really important right now as we move to these younger cohorts for us to really be armed with the right knowledge to have these conversations with our patients. So to warm us up in the usual webinar style, we've got a poll for you because we'd like to see just how many people you have been engaging with. So Simon, if I could just ask you to bring up the first poll, and I'll give you a few seconds to lodge your votes.
Of course, hopefully you should see that now.
Great. So, you know, so many of us are already having conversations with people who are showing signs of potential hesitancy because they're asking the questions, the right questions about the safety, the efficacy, the religious acceptability of COVID vaccination. So that's really, really interesting. Are we able just to launch the second poll just to see where people's confidence is with giving this sort of advice to patients?
Of course, I'll do that now. There you go. Okay, so this poll's made up of 4 questions, just multiple choice there. So if we can encourage you to do that, please.
We really are testing you tonight, aren't you? After a long day at work. 4 questions. Sorry, guys. Okay, so varying levels of confidence on the different topics. So hopefully after today's presentations from Dr. Shirley Hopper and Professor Jeremy Brown, we should all be hopefully feeling a lot more confident with our answers to sort of move forward and go on and have these conversations with our patients. So without further ado, I'd like to introduce Dr. Shirley Hopper, who works for the MHRA, to start her presentation. Thank you, Shirley.
Thank you very much, Rena. Yes, as Rena said, I'm Shirley Hopper. I'm a medical assessor at the MHRA, and I am part of the team that has been assessing the COVID-19 vaccines. I've been working at the MHRA for about 12 years, and before that, I was in general practice. So if I can have the next slide, please. Okay, so as I'm sure you're all aware, there are currently 3 COVID-19 vaccines approved in the UK, and the first 2, Pfizer, BioNTech, and the Astrazeneca vaccines are the ones that are actually currently being rolled out in the UK, and the Moderna vaccine is expected to arrive in the next month or 2. The Pfizer and the Moderna vaccines are both mRNA vaccines. They have quite a similar, although not identical, platform. They're both comprised of the genetic material that codes for part or all of the spike protein of the SARS-CoV-2 virus, and this is encapsulated in lipid nanoparticles. So what happens when it's injected into the muscle is that the lipid allows the RNA to enter our cells, where it then encodes the spike protein, which is expressed on the surface of the cells, and then this causes an immune response to the spike protein. The Astrazeneca vaccine is a bit different because this is actually an adenovirus which has been modified so that it actually makes the spike protein and expresses this on its surface. So after injection, the adenovirus, which is actually a chimp adenovirus in this case, which is modified so that it can't replicate in the body, this is then taken up by our cells, and then the spike protein is made and expressed on the surface of our cells. So in the same way, the immune response is to generate immunity against the S protein, and I'll say a little bit more about that in a minute. So as you can see, we actually started looking at the data on these vaccines well before those headline results were published in the newspapers. And just as a reminder, I've included also the storage temperatures. So as you're aware, the Pfizer and Moderna vaccines are frozen, whereas the AstraZeneca one is refrigerated. Next slide, please. While we're just waiting for the next, here we are. So this actually just gives you an idea from the vaccine developer viewpoint. It's the normal development that we see for all medicines and vaccines, but it is compressed because of the pandemic. So, I mean, when you think about it, it's amazing actually what's been achieved in a short space of time, from the preclinical studies through toxicology studies, and then getting clinical trials authorized, and then the normal phase 1, phase 2, phase 3 stages prior to approval are compressed and overlapping. And also the companies have actually started production even though they didn't know whether the trials were going to be successful. So they have taken a risk with this. And then they've actually then sent their data in to the regulators while the trials are ongoing, so that we can get on and start assessing, so we can be really prepared for when the final data is available. Next slide, please. So at the MHRA, we tried to get prepared really early. Obviously, in the summer, we didn't know if these vaccines were going to be successful. But we got together a working group of experts to advise the Commission on Human Medicines, which is our statutory body, which makes recommendations on medicines at the MHRA. So the expert working group is made up of experts on vaccines that would then be able to provide this advice when needed. In addition, we started preparing for independent batch testing, which has to be done for vaccines. And we also started looking at the pharmaceutical and the preclinical aspects because this data was generated earlier. And even for the clinical analysis that I was involved in, we started looking at trial protocols, assessing the methodology, started writing our report so that we were ready for when the results came. Next slide, please. So in this way, we were able to actually do the final assessment quickly without cutting any corners. So I'm going to just talk more obviously about the clinical data because that's my area of expertise. And this is divided for vaccines into three components: immunogenicity, efficacy, and safety. Immunogenicity data for the vaccines has been generated in phase 1 and phase 2, although sponsors are testing for immunogenicity in phase 3 as well, and we'll get that data later on. I'm just aware that the sound quality is not that good, so obviously please let me know if it's a general problem, please. And then
I can hear you clearly. Sorry, Shirley. You can hear you.
Okay, yeah, I just got a message flashed up.
Oh, okay.
Okay. And then the efficacy is really in phase 3, and I'll explain why a bit later. And then safety, we look at that through all phases. So even in a small phase 1 study with a few volunteers, we would very carefully look for any evidence of any side effects. Next slide, please. So the immunogenicity, this is really the immune response to the vaccine that we're measuring in humans. So these studies were conducted in healthy adult subjects, some were up to age 85 years of age. And there's two parts to this. There's the humoral or antibody response, and that's measured by levels of antibodies that bind to the spike protein. Also, a different test for neutralizing antibodies. So these are antibodies that can actually stop the virus entering cells. So that's a more functional measurement. And then these levels are compared with samples of serum from patients who are recovering from COVID-19. So we were able to make sure that the immune response to the vaccine was at least as good as the vaccine in convalescent patients. And then we're also very interested in the cellular response, and it's mainly T cells that have been tested to see whether they actually do respond to virus proteins. We were pleased to see a strong immune response also in subjects over 65 years of age, and that gives us reassurance about efficacy. Now, we know that as people get older, the immune response does diminish, and we did see that in the studies, but it was still comparable to the convalescent serum. Next slide, please. So the efficacy, we were looking for data from well-designed studies in tens of thousands of participants. And there were guidelines from WHO and also from the FDA that were put out to industry at an early stage. And as a result, we did see very well-designed studies. And the primary endpoint for all the 3 vaccines was the incidence of COVID-19. So what that means is a symptomatic SARS-CoV-2 infection, but it doesn't have to be a severe infection. It can be a relatively mild illness. And this has to be confirmed by PCR. So obviously nasopharyngeal swabs were taken, and also confirmed by by a blinded adjudication committee. So the way that these studies are designed is that there's as much blinding as possible. So the recipients are blinded, and the sponsor staff that are dealing with the recipients are also blinded. And only the sort of statisticians dealing with the data and the people who are actually preparing the vaccines are unblinded. But as an additional layer of robustness, all the cases were adjudicated as well. I should also mention that these were placebo-controlled trials in most cases, apart from some of the Astrazeneca ones, which were compared against the meningitis vaccine in a randomized way so that we could compare against background incidence. So this measurement of incidence of COVID-19 had to start at least 7 days after completion of the course to give the immune response time to kick in. And we were interested for the primary endpoint in the participants who had no evidence at baseline that they had had infection. Now, there was about 3% in the trials who did have evidence of infection, and these were kept in for safety, but excluded from the efficacy analysis. And obviously, we were also very interested in looking at the severe cases, the hospitalizations, and also asymptomatic infection, because that gives us an idea of transmission. So the way that we measure efficacy, feedback. Well, that's better.
Sorry, I just had to mute somebody there, sorry.
So the way that we measure efficacy is 1 minus the relative risk. So for example, if in the vaccine arm, the incidence is 25% of that in the placebo arm, then that would translate to a vaccine efficacy of 75%. So all 3 vaccines did have good efficacy, as I'm sure you're aware, and the point estimate of vaccine efficacy for this primary endpoint of symptomatic disease was between 70 and 95%, depending which vaccine we were looking at. Now, this was well above the WHO target of 50%. And also, these studies were very well powered. They were very large studies. So the precision of the estimate, the confidence intervals was also good. And there was also evidence of efficacy against severe disease, hospitalizations, and also in clinically important subgroups, such as those who have risk factors for severe COVID, and also in older subgroups. Although for the Astrazeneca vaccine, there was less evidence in the over 65s, and that was simply because they had less subjects in the over 65s. But we were confident of the efficacy in this age group because we had good immune response data as well. And we are expecting to get confirmatory efficacy data in the next few weeks, and we'll update all our information about that. But we felt confident enough based on the immune response. Next slide, please. So moving on to the safety, we requested, and all the other regulators also requested, an average of 2 months of follow-up, so a median, after completion of the COVID-19 vaccine course, which obviously for these 3 vaccines, which are 2-dose regimens, it was at least 2 months of follow-up after the 2nd dose. Now, the safety data is very robust because of the size of the studies and also the comparison with control. So compared to a lot of medicines that I look at, you know, this was very large numbers to look at, but made us very confident that we had a good characterization of the safety profile. Now, in all three vaccines, the trial subjects were given electronic diaries so that they could record the local and systemic reactions that are usually seen with vaccines for 7 days after each dose. And this showed that most frequently, subjects got pain or tenderness at the injection site and also flu-like symptoms. Now, these are usually mild or moderate in severity and resolve after 2 to 3 days. And they are more frequent in the younger participants that Rena was talking about, the vaccine being rolled out too soon. And that's obviously something to be aware of. So you may want to advise paracetamol if side effects are troublesome. And you may want to point this out to younger participants that then they may not feel like going to work for a day or two after the vaccine. I think it's better to warn people about that. And it's also interesting with the vaccines that for the Astrazeneca, the reactions were actually more severe after the first dose than the second dose, whereas for the 2 mRNA vaccines, it was the other way around. The trials also look for any adverse event. So that's not necessarily a side effect, but anything that happens, even a road traffic accident, would be included. So serious events, deaths, medically attended events, that was all recorded. And obviously with the control arms, we could get a good idea about whether there was anything to worry about. And we didn't identify any major safety issues for these vaccines. And there was also no indication of a worse safety profile in people who had been positive at baseline for infection with SARS-CoV-2. Now, all these studies are planning 1 to 2 year follow-up. So we are going to get data coming in. We'll obviously update our information. Next slide, please. So moving on to post-approval. As I said earlier, a bit about trial populations. So as you'd expect, these trials included men and women, roughly 50-50, people from different ethnic backgrounds, younger and older adults. The oldest, certainly in the Pfizer studies, was 89 years, and also people with stable medical conditions. So the Pfizer-BioNTech trials took place in the US, in Europe, South Africa, South America. As you'd expect, from those locations, the participants were mainly white, about 10% Black or African American and 4% Asian, and nearly a quarter over 65. AstraZeneca trials in UK, South Africa, Brazil, also a similar spread of ethnicities. As I said earlier, less were over 65 years of age, so we don't have that precision for the efficacy in the older subjects. And the Moderna trials were in the US, so again, similar ethnic profile and about a quarter over 65 years of age, so quite similar to the Pfizer study. Next slide, please. So the next slide is about the benefit risk. So as I said before, this expert working group that's been set up evaluated the data alongside assessors such as myself in real time. So sometimes they met twice a week to look at the data with us. And based on their advice, the CHM recommended that all 3 vaccines should be authorized. Now, there is a relatively short duration of trial follow-up of median of 2 months. And this is less than we would normally accept for a vaccine. But this needed to be balanced against the public health need for COVID-19 vaccines. And it's also worth bearing in mind that for vaccines, generally serious effects are seen within the first 6 weeks. So that's why the 2-month average was chosen by regulators. And also manufacturers will have to submit their longer term data, and that is a condition of the authorization. Now, there is limited information on use in pregnancy, There have been pregnancies in the trials, but we don't have the outcome data yet. So that's why administration in pregnancy should only be considered when the potential benefit outweighs any potential risks for the mother and for the fetus. So Professor Brown may say a little bit more about that later, talking about the JCVI recommendations on that. But there's no evidence at all that vaccines affect fertility. Next slide, please. So after approval, we test every batch at the MHRA independently in parallel with the company's testing, and we're continuously monitoring vaccines post-approval to detect safety issues. And this includes any very rare events that wouldn't have shown up in trials of tens of thousands of people. And we are receiving on average about 3 reports per 1,000 doses at the moment, which is higher than we normally get for vaccines, but perhaps that's expected. And most of these are the reactogenicity events, the painful arm, localized reactions, and the flu-like reactions that I described earlier. Now, I'm sure you will remember that very soon after the Pfizer vaccine was approved, there were isolated severe allergic reactions reported during the rollout. And overall, it looks like there's about 1 to 2 cases per 100,000 doses of Pfizer vaccine. And in the US, where they also are using the Moderna vaccine, they've seen a similar rate. So once it settled down and everybody's done the calculations, it looks like severe allergic reactions are very rare. But as a precaution, people who receive the Pfizer or Moderna vaccine should be monitored for at least 15 minutes afterwards. And this does not apply to the Astrazeneca vaccine because we haven't seen these severe allergic reactions at all. Although, of course, there is always the theoretical risk, and there are warnings about that in the product information. And all 3 vaccines are contraindicated in people with a previous history of severe allergic reactions to any of the ingredients of the vaccine. And this, of course, would include someone who had had a severe allergic reaction to the first dose, such as generalized urticaria, and they would not be offered the second dose. So following the very substantial exposure, no other new safety concerns have been identified so far. Next slide, please. Yes, so we have information for healthcare professionals, so obviously that includes yourselves, and this is equivalent to the SMPC, but because this is actually, these vaccines are approved under Regulation 174, which is different to a routine approval. We don't actually have an SMPC, but it's a very similar format. And we have information for UK recipients, which is equivalent to the pill, and that's based on the SMPC type document. We also have published public assessment reports. We've just published the one for Moderna, and they're available on our website. And they give some more details about how the vaccines were assessed and a bit more background to the clinical trial data that I've summarised. And of course, we encourage healthcare professionals and members of the public to submit any suspected side effects through our yellow card scheme. So this link will give you access to the yellow card scheme, but also gives you links to the product information and the public assessment reports for all 3 vaccines. So just want to thank you very much for your attention, and happy to answer questions. Thank you.
Thank you, Shirley. That was really good. I love the science, and it's nice to revise that immunology again, isn't it? And think about all the trials and how they've been set up. So that's really, really useful, and actually very specific information that we can share with patients on, you know, the numbers of side effects that people are getting. I know the allergy one I've been asked about a lot because I've got a lot of patients with medication allergies and sort of very, very concerned. So that's definitely one that I've been coming across in my pharmacy. We've had a few questions, but if that's okay, I'm going to leave them towards the end, because we'll see if Jeremy can maybe cover some of those questions. But also, please do keep your questions coming in. I think one of the questions is will your slides be available? And we are recording this webinar, so the whole webinar is being recorded and will be shared on the MPA website. And if you've registered today, you'll be sent a recording of that automatically for this evening as well. So without further ado, Professor Jeremy Brown, if you'd like to just let us know about your thoughts from the JCVI point of view.
All right, thank you very much, Rina. So just some background about myself. I'm an academic at University College London who does quite a lot of vaccine type research, and respiratory infections is my thing. I'm also a clinician, so I've been on the wards looking after patients with COVID during the first wave and the second wave, because I'm a respiratory physician who specializes in lung infections. So, you know, this is my baby really when it comes down to it on the wards. So who do we give the vaccine to and why? So that this was relatively straightforward decision-making process by the JCVI. With the first wave, about 50,000 people died. Second wave is another 50, 60,000 people have died. And the vaccine is being deployed to prevent death. That is what it's for. It's not to prevent infection, it's to prevent people dying from the infection. Now, obviously, if you prevent infection, you will prevent them from dying from infection. But the end point that we're looking at is people going into hospital and having severe COVID. And that's the whole plan for the first phase of the vaccination program. So who dies from COVID? Well, you're probably very much aware of this, that it's all related, well, most of it's related to age. If you're living in a nursing home, you have a 20-30% mortality to catch COVID. If you're over 80, it's 10%. If you're over 70, it's 2.5%. If you're over 60, it's about 0.5, 1%. And then it rapidly tails off. So that if you're under 30, the chance of dying of COVID is 1 in 50,000 or something along those lines. It's incredibly rare. So that makes it very clear and very straightforward to identify those who need to be vaccinated in the first wave. And that is on this age-based process that you're well aware of, where we started with the over 80s, moved on to the over 75s, then the 70s, then the 65s, and so on, down to age 50. And that's the end of the first wave. Within the first wave, we've also included other people who are high risk of mortality and severe disease, and those with underlying medical disorders. And there's two categories. There's those who are very, very at risk, and a lot of those are largely to do with cancer and being immunosuppressed, although Down syndrome is in there as well, and people with severe cardiorespiratory diseases, etc. And then there's another bunch who are with less severe comorbidities, who are placed in the sort of 60, 65-year-old age bracket parallel with that. So that's the first wave. The second wave, the second phase of vaccination, I'm not going to talk about. That's being decided at the moment and is a bit more complex and doesn't kick in until we've got all the people that are vaccinated down to the age of 50, which is actually a lot of people. That's like 25 million people. It's pretty close to 50% of the whole adult population. So that is no insignificant milestone. That is a massive success once we get to that stage. And that is aimed to be in April, towards the end of April. So that's who we're vaccinating and why we're vaccinating them. The other fairly controversial decision that we made very early around Christmas time was that we should delay the second dose of the vaccine. Now, I actually am horribly surprised at how controversial this decision is, because if you look at the data, it's an absolute no-brainer. You give one vaccine and the protective efficacy of the vaccination is given and provided by that about 80% of that protective efficacy is provided by the first vaccine. So, for example, with the Pfizer vaccine, what happened between the third and fourth week in the trial is that 90% of the infections that could occur occurred in the control group and not in the vaccinated group, given a very good vaccine efficacy for one dose. Then they get their second dose because that's the trial was set up to do in the third week. sorry, I spent the second and the third week. And the third week, they get the second dose, and thereafter, you can't tell the effectiveness of one dose. But the point is that one dose prevented most of the disease that was occurring in the second and third week, which is the earliest you might expect that one dose to kick in and have its vaccine efficacy. The data for AstraZeneca was much better in that there was a bigger spread between the first and the second vaccine, up to 8 weeks between the two, varying between different groups of people due to vaccine supply issues when they set the trial up and were doing the trial. And that allowed us to identify that, in fact, delaying the second dose doesn't weaken the efficacy, it improves the efficacy of the vaccine, and it also strengthens the antibody response you get when you get given your second vaccine dose. And of course, if you give 2 doses of vaccine to one person in 4 weeks, that means one other person isn't getting a vaccine at all. And in the midst of January, when we had literally tens of thousands of people coming into hospital with COVID, the absolute necessity was to vaccinate as many people as quickly as possible. And as one vaccine gives 80% of the protective efficacy, then it's quite clear that one dose and a delay for the next dose, 2 or 3 months, and come in with the second dose later on is the most obvious thing to do. So that's why you went for the delayed second dose, because it allowed us to get as many people as possible protected. The protective efficacy from one dose was the majority of the protective efficacy you get from vaccination anyway, and delaying the second dose is a positive thing because it improves your antibody responses later on by 2 or 3-fold. So that's why we had the delayed policy. Safety, Shirley has described it in detail and given all the statistics. The one thing I just want to remind people is that since the trials, we have now vaccinated 15 million people in this country. Half of Israel have been vaccinated, and a very high proportion, well, not very high proportion, but 10% of America has been vaccinated, something like that. And that is gazillions and millions of people. It adds up to a very large number. And if there was a significant safety signal of a severe complication occurring, we would know about it by now. Essentially, the safety issue is twofold. One is the anaphylaxis issue that Shirley mentioned, which is very rare. And the other is actually you do get quite a strong flu-like reaction and local inflammation from the vaccines. And that is a bit more than you could get with normal vaccines, probably. And it is worth warning people who have been vaccinated that it is possible they're going to be off for a day or two feeling a bit grotty. But it's not long-lived and it's not serious. It goes away by itself. And it's not COVID. There's always after the vaccine, in people who've just been vaccinated, in the first week after being vaccinated, there's a big increase in people being tested for COVID because they think they've got COVID because they've got a bit of a fever and feel a bit grotty. And that's just the vaccine response. Okay. I'm not going to talk about types of vaccine because Shirley has covered that. Just to say that there is no real difference in efficacy between the vaccines for the endpoint that we want, which is protection against being admitted to hospital and severe disease. I will talk just a little bit about the alternative vaccine strategy that you can have, which is that for many vaccines, and this is like a good example, is the flu vaccine in children. We use flu vaccination in children, not really to prevent children from getting a flu, but to prevent the children giving the flu to their grandparents. And that is where you're vaccinating a group of people to prevent spread of disease. And we didn't consider this as an option for COVID because the trials did not tell us whether the vaccines would prevent spread of disease from individuals getting infected at all. So we had no data. Also, we weren't entirely sure who was spreading disease, and we're still a bit unclear about that. So there's 2 good reasons why that strategy has not been taken on board. It's becoming clearer that the vaccination is having a good effect on preventing disease, even in asymptomatic people. And if it does that, then it would prevent spread of disease because far few people will be able to catch it from anybody else, from an infected person. So if you're vaccinated, the chance of catching disease will go down a lot. And that means that you're not going to spread and the R number will go down as a consequence of vaccination of more and more people in the population. So for phase 2, that can be brought into the equation and thought about and discussed, but it's not the rationale at the moment. Now, there's two more points I want to make. The first one is, right, okay, why do we need to get a high uptake of this vaccine? The way I think about this, and it's a little simplistic, but each of the two waves that we've had, about 10% of the population have been infected and it's killed 50,000 people. If we have a vaccine uptake of 90% and the vaccine efficacy is 90%, that means 20% of the population can still get disease. That means there can still be two more waves of 50,000 deaths. Now, the vaccine efficacy at preventing severe disease is probably much higher than 90%. We would hope it's more like 95%, but we don't know. We haven't got the data yet. But it becomes essential that as many people who are at high risk of disease as possible are vaccinated. As I said before, that is age, but to bring in a few other things, ethnicity, male sex, low economic class, living in a large household with lots of multi-generational people staying in the same household, certain jobs, public-facing jobs, they are all risk factors for catching COVID and mortality. And the higher proportion of the population that are unvaccinated in the really risk population, the ones I've just mentioned, the more likely it is that there will be a wave which will kill a lot of people despite high vaccination. So you could vaccinate 100% of people who are white living in houses in Surrey. And if you've only vaccinated 50% of people who are living in an inner city, I don't know, Lewisham, Hackney, where I live, Islington, then you are still going to have a horrendous wave of infection. So it is incredibly important that the uptake of this vaccine is high in those who are at risk of this disease, because otherwise it will come back again. Even though you may have vaccinated 80% of the population, if that 20% of unvaccinated people is concentrated in those who are at high risk of infection, then it will be a major issue still. And I think pharmacies and community pharmacists, with your outreach and your knowledge of the local population, are an incredibly important arm in our weaponry to make sure that the vaccine uptake is high in the populations that really do need it. You will have better connection to your local community than, say, the big vaccine centres have. And you'll be much better, I hope, at achieving high uptake in those who at present seem to have a degree of distrust of vaccination or can't access vaccination for whatever reason. And then the last point I want to make is that we're not getting out of this COVID pandemic until everybody in this country is vaccinated. All right. It is going to be coming back and being a problem in vaccinated adults in the country. And we cannot just hope it's going to go away. This is going to have to be an active process of really supporting people we can. This huge hole that we're in caused by this pandemic. Thank you, and I'm very happy to answer any questions that you may have.
Thank you, Jeremy. Now, if that wasn't an inspiring call to action to all of us to take our part in really supporting people to get the vaccine, I don't know what is. I think that's really hit home to me about how important our role is in proactively having those conversations with people coming into our pharmacy, because you're absolutely right. We need to really make sure that everybody gets this vaccine so that we are all safe. Otherwise, we will be having these lockdown situations and more vulnerable people will be getting ill and sadly dying as well. We have had a few questions through on the Q&A. Please keep them coming if you're attending. I really would appreciate your questions and I really would like to answer them. So the first question we've had from Farah, which I was going to ask Shirley if that's okay, is part of the trial data. A lot of the patients who were in the trials obviously weren't from Asian and Black backgrounds, and that's possibly causing some of the hesitancy within those communities because they feel like this was tested in people that aren't like them. What's your response to that?
Okay, so overall, it was about 10% Black and about 4% Asian. Yes, I take the point, but it's worth bearing in mind that these were really large trials. I mean, they were tens of thousands of people in the trials. And so, you know, the numbers from Black or Asian backgrounds are equal to the numbers that we often see in a whole trial for other medicines. So we do have, you know, we do have the evidence, even though the percentages appear small just because of the huge numbers. And also there's no mechanistic reason at all why these vaccines shouldn't work just as well in people from BAME groups.
Thank you. And I think that was going to be the next question. What's a normal kind of profile of trials? And actually, that's probably another call to action, isn't it? To get more BAME people to be encouraged to join clinical trials so that we don't keep asking ourselves these questions. And actually, you're absolutely right. Medically, is there anything that different to us that will make us respond? And over many, many years, we've had vaccines throughout all communities and cultures. And, you know, we haven't seen a different response, have we?
There is,
Jeremy, did you want?
Yeah, there's no difference in vaccine responses with ethnicity. You have to bear in mind that there's more genetic variation within Africa than there is outside of Africa. So an Asian person and a white person is closer genetically than most African populations are to each other. So you have to be, you know, it isn't like, yeah. So the genetic variation issue is bigger within Black Africans than it is outside. So it's not affecting it. The trials in South America, 30% were mixed race. I mean, it was a very high proportion in Brazil, as you might expect.
And just to add, because we did have 10% Black subjects in some of the trials, we were able to do a subgroup analysis, which actually did show vaccine efficacy independently in that subgroup with good precision. That wasn't so easy with the 4% Asian, but taking on Jeremy's point about the genetic diversity in Africa, I think that 10% subgroup analysis should provide reassurance.
And I guess I saw you mention, Shirley, that there's a 1 to 2 year follow-up that we're kind of involved in at the moment. So are BAME communities particularly being followed up so that we can, you know, just prove that it's safe for these communities that may be hesitant, for example? And also just thinking about, you know, is 1 to 2 years the norm for vaccines? I've got a lot of people sort of saying, what if this affects my children's children? You know,
okay, so when
and people really concerned about really.
Sure. So when I'm talking about the 1 to 2 year follow-up, that is in the studies themselves. So, for example, in the Pfizer study, there were 44,000 participants in that study, and the aim is to follow all of them up for 1 to 2 years. So they will be having repeat immunogenicity testing and be monitored for safety and efficacy. So the genetic profile of that trial isn't going to change because they're not going to recruit more people to that trial. They're simply going to follow up the people that started in that trial because the trials are no longer recruiting. So really what we're looking at is real world data now to look at more effectiveness and efficacy, and that's being done by Public Health England and other bodies to actually follow up and see in the real world does the vaccine reduce the risks, and we are now seeing that. We have now got the data. Great.
Another question that we're getting asked a lot, definitely in our pharmacies, obviously there's the question about the BAME populations, but also there's a religious aspect, and a lot of people feeling that it goes against their religion maybe to have vaccinations, or that they're worried that there are certain animal products in these that would be against their religions. Is there any sort of advice that you'd give in that context?
Yeah, so none of the 3 vaccines that I've been talking about contain animal products.
That's great. And I think there's been a lot of community leaders of various faiths. I've seen them lots on TV and across social promoting that these vaccinations are safe and approved, and people are encouraged to have these vaccinations. So that's been really, really nice to see, isn't it? Another question, is there any evidence that side effects can last longer than a few days? And I noted both of you sort of talked about being really honest with patients about that side effect risk, and I think that's really, really important actually, because like I said, we don't want to convince people you'll be okay and just have it I think it's really important to be open and honest about the side effects. So has there been any evidence of these side effects lasting longer than a few days?
Yeah, so as I said, what they did was they did this sort of e-diaries for 7 days, and vast majority of the side effects that I talked about, the local and systemic flu-like effects, were over well within the 7 days. There was a small minority where they did persist, but I mean, you know, that was really a minority, and we didn't see people suddenly getting side effects after 2 weeks. So yes, in some people, you know, the reactogenicity was a bit prolonged, but in the vast majority, it was over within 2 to 3 days. And we don't suddenly see someone getting a side effect popping up at 2 weeks or 4 weeks.
Okay, that's good to know, because that's going to help us to reassure patients. A question to Jeremy. Oh, sorry, Jeremy, you wanted to come in there.
Well, I just want to say that it's an important point to make here is that the vaccine constituents are gone. They disappear after a few days and a couple of weeks. They just cannot persist within the body. They'll be broken down and gone very quickly. So the idea that they might persist and cause problems long term is just, it's not scientifically at all likely.
Yes, and just to add to that, the vaccine, the whole point of the vaccine is to try and induce the state that you would be in had you had COVID, without having all the danger of having COVID. So you're left with the immunity without all the risks of COVID. So that's why we can be confident that there's no effect on fertility, because there has been some discussion, I think, about you know, antibodies to S protein being an issue for fertility. But of course, this is what you see when you've had COVID, and we've got no evidence that COVID itself is causing any problems at all.
Okay, that's good to know. There's a question here probably for Jeremy. Must the follow-up vaccine dose be the same type as the first? So I assume you also mean if you get the Pfizer first, can you have the Astrazeneca second? And what's the effect on efficacy? I'm sure that's a hot question you're being asked quite a lot.
Well, I mean, the bottom line is that you should get the same vaccine for the second dose. But I can see in the future that if there's a third or a fourth vaccine required later on, a booster, it's not likely to be the same one as the first one. In theory, there is no real issue with having a mixed vaccine, having a Pfizer then an AstraZeneca. They both have the spike protein as their antigen. They both target the same thing. It's just the delivery system is different for AstraZeneca versus the Pfizer. And the Pfizer and the Moderna delivery system is very, very similar. So no, you shouldn't be getting doses which are different, but if you did, I can't see it being an issue, really. I mean, it's going to generate the same antibody response. There's a theoretical reasons why it may be better. And in fact, there's a trial going on now where they're mixing the doses. They're doing a Pfizer, then an AstraZeneca, and they're doing an AstraZeneca, then a Pfizer, to see what difference it makes. And I suspect that trial will show very little difference between them, or subtle difference of great interest to academics, but not to practical reasons.
I can see it. Yeah, I can see a theme of questions. A couple of people asking about the vaccines being vegan, vegetarian, the ethical aspects of fetal cells being used in the process of the AstraZeneca vaccine. And I think you mentioned chimpanzee RNA being used. So any sort of thoughts on that? Because funnily enough, before I joined this, I was on a local patient support webinar run by Public Health. And those questions are coming up there actually by members of the public as well, very frightened once they
yeah, so I can start. Jeremy might want to come in, but yeah, so the chimpanzee, it's not anything that's derived from chimpanzee. It's an adenovirus, which, you know, in humans can cause a cold type illness. The reason I think they chose the chimpanzee version of the adenovirus is because humans are less likely to have immunity to it already. And the adenovirus has been altered so that it can't replicate in the body anyway. So it's just broken down and disappears. It is just a vector, and we have other products that use a vector. So the fetal cells, yes. So the AstraZeneca vaccine is grown in in a fetal cell line. That's how it's produced. Yes, it is derived from human fetus cells, but this was from a very long time ago. You know, these cell lines are used to produce a lot of vaccines, and there's no trace of the fetal cells which are removed during the production process. And we do have some statements from religious leaders on the acceptance of this.
Yeah, it was isolated in 72, the fetal cell line that's used, and the cells have been replicated so many times since then that they're just, you know, it's like hundreds of generations away from the original isolation. And they are vegan. I mean, there's no animal products in these at all, or milk-based products. No. Yeah.
Thank you. There's been a few questions I think we've already covered in the webinar, so I will type in the answers to people who've been asking some of those questions. Just finally, a question to both of you. Will we be looking at revaccinating this winter, considering variants, and will it be a sort of an annual thing that people are sort of hinting at? Any comments on that before we move on to our panel discussion?
I guess that's for me, and the answer is we don't know. So there'll be two reasons for revaccinating. Okay, so, and I think, so first reason is that the first vaccine at some point will wear off in its efficacy and you need to be revaccinated just to boost that again. And we have no idea how long it will last. The protection that you get from the vaccine is pretty good. So that normally means the vaccine efficacy is going to last a few years. So that wouldn't necessarily make us want to revaccinate in winter. But we don't know. So we have to keep an eye on the immune response to the vaccine in people over time to see what sort of rate it's waning, it's getting weaker at. And the second reason is the new variants. If there's a new variant that comes around that the vaccine is not that effective against, then we need to revaccinate with a new vaccine. Well, I say a new vaccine, a vaccine that has been modified to contain that new variant. And that's relatively straightforward to do. You know, you can do it in the laboratory in a few days. It's convincing Shirley that the new vaccine is good enough is the bit that takes a bit of time, but it's actually relatively straightforward to do that, and that there isn't good evidence that we need to do that at the moment, because all the data that we're getting with the vaccine in England and Wales and Scotland is actually with the Kent variant, because that's the vaccine's been used at the same time as the Kent variant has become dominant. So we know from the data that we have, and that was released yesterday, yeah, that the vaccine works against the Kent variant. So that's good. That means you don't have to revaccinate against the Kent variant in the near future. The South African variant, the Brazilian variant, and future variants that will crop up, we have to keep an eye on. And if there are some that do escape our vaccine, then we will have to revaccinate, but we just can't tell at the moment. So there's a possibility of revaccination. I mean, I think people have mentioned the autumn doing it again. To be honest, I think it's unlikely we're going to have to do it again this year. But yeah, it's something we're thinking about and looking at.
And the one question I was going to ask you that you said you're not allowed to talk about what happens to the under 50s, but we'll maybe have to host another.
Can't talk about that. Not allowed to. I've been
webinar when you release that information from the JCVI. So thank you. There are some questions still in the Q&A, and I'm going to ask Shirley and Jeremy if they're okay to start replying to some of them. I think some of them we have covered in the presentations, and they may just have been missed in terms of factual data. So now moving on to my two esteemed colleagues, Bola and Ade, who are some of my favorite community pharmacists out there, and I know extremely passionate about speaking to patients, really promoting the COVID vaccinations, both with walking into their pharmacies, but both of them are also directly involved in providing COVID vaccinations to the public as well. So welcome to both of you. So what do you think, what sort of people are coming into your pharmacies, and what are the sort of questions they're asking, and how are you responding, and how are your teams responding to these questions? Bola first, if that's okay.
Hi, so thank you very much. It's such a lovely and very informative webinar. So thanks for hosting this to the MPA. So I have two roles, and I'll briefly describe these roles. So one is as a community pharmacist in West Croydon in London, where majority, and when I say majority, that's up to about 90% of our population are BAME. My other role is as a CCG pharmacist, where I've been heavily involved in the rollout of the program. So setting up the vaccination sites, ensuring that the clinical governance and vaccination oversight, so around the cold chain storage, are in line with the guidance and the protocol. So I've been involved from, different angles. So in the type of patients that we've been seeing is really very broad, and people wanting to know more about the vaccine in order for them to make an informed decision. A typical example, people will come in, they'll ask questions, they've heard all sorts of things through the social media, members of their family, and sometimes stories or incidences that have changed over time. And you probably remember the incidences of a couple of healthcare professionals who had a reaction to the Pfizer vaccine. And you'd hear stories about, them dying. But actually, no, they didn't die. They had a severe reaction and they recovered from that reaction. So it's important as pharmacists that we actually are aware of these facts, which is why we're here today, to be able to reassure our patients. But most importantly, this is our forte. This is where, we can use our experience of talking to patients in very simple language. And we're used to people coming in, asking about their medicines, even medicines that are not on their, that are not prescribed for them, medicines they've come across on the internet, medicines that have been offered to them by friends or family. And it's in the same vein that if we, if we are up to date with information around the vaccines, we can reassure them. And just quickly, so I can have Ade have the say as well, I'll just cite an example of a lady who came in. And she came in for completely something else. And then she just started asking questions about the vaccine. And for every question and for every answer you provided, she had another question and she wasn't convinced. Knowing this lady, and I looked at her and I said, do you know anyone who's had the corona infection? She said yes, her neighbour and somebody else who knew somebody else. So I asked her a simple question: what would you rather have, the corona infection itself or the vaccine? And she looked at me and she went, that's another way of looking at it. She had never thought of it from that angle. So sometimes it's just about giving them or jugging something, letting off a light bulb moment. And I wasn't expecting her to give me an answer, but for her to go away and reflect on all the information she'd heard and to make a decision.
Thank you, Bola. And I think you've put it beautifully there, isn't it? Our job is to get people to have that light bulb moment of being informed, having the facts, having the honesty as well, which I think has really come out from today's webinar as well, to just be honest with patients. I know when I had it, I did react to the vaccine, and I've been really honest with my patients who've come in and said, yes, I did have side effects. It lasted about 12 hours. I felt rough and rotten, but I recovered, and it surely is better than having COVID. And that's the sort of message that I've been saying to my patients as well. So totally, I love that light bulb analogy. Ade, I'm sure you're equally inspiring and have been supporting so many in your role as well. And I wanted to sort of bring in some of the questions that have been coming in as well about the social media sort of disinformation or misinformation that's really been spreading about the COVID vaccination, and that one person who died a few days after they had the vaccine, it must have been that. It goes around, spreads like wildfire, doesn't it? But the thousands that have been saved that from having the vaccine doesn't go around social media, does it? So what are your thoughts on that, Ade?
And I think just to thank Bola for what she just shared, and very much, I guess, my role, as well as just being part of a contractor that is actually helping to provide the vaccination in my local community, is that I'm also the Public Health England pharmacy champion for vaccinations, which involves a whole range of different activity around just educating and building and increasing awareness of the value of that. But one of the things as well is that I do get quite a lot of opportunities to do a lot of things in the media. And I have deliberately actually tried to position myself as a kind of ask me a question role in this, because I, like everybody else, has actually had to go through a journey of looking at this and understanding, okay, how does this work? What are the questions that I have, and what are the questions that people have had? And even for my team, I remember the journey here was actually for them telling me why they didn't want the vaccine a few weeks ago and months ago. And that journey of, okay, tell me why, and the questions and the conversations, and actually just giving people the space to have that conversation, which is something that community pharmacy is uniquely able to do, because we do have this great trust dividend in our community. And actually, you can come in, you can ask us questions, we can give you opinions, you can go and search it up, you can come back again. But ultimately, one of the most important questions that I keep having people ask me is, have you had it yourself? And I think I can't stress enough to colleagues that actually not just being able to answer yes to that. And I know that, NHS England has really launched a campaign, which I also have to support, about why pharmacy colleagues should have the vaccination, but also actually not just saying I've had it, but also telling people why you've had it, and actually your own journey. Because like Bola shared, there is those human connections there. There is this thing of, actually, is this going to be safe for me? And is this something that I need to be worried about? Or there, because you can spend a lot of time trying to deal with the, disinformation on WhatsApp and, social media. But ultimately, those things morph into new things, and there'll be new things, and there'll be. But what you can do is you can actually use that great trust that people have in you to say, well, I've had it, and my family's going to have it when their time comes. And this is why, because even though we weighed all of these things, and then I just, I thought about my friend that had COVID, and I saw him, or then I thought, oh, I lost a relative, and immediately people connect with that, and that's it. And the joy of it is that whenever you get one person who has it, they immediately become a great campaigner, because what they would do is they'll go back to the same people that they've had in their bubble who have been hesitant and go, well, I've had it. And look, the lady said I would be unwell for maybe a day or two. I might have a bit of temperature, my hand may hurt a bit, but actually after that, I was fine. And I think, you know what? That is the great role that we have in this. I think it's also important that we are part of the program as well, because that allows us to have a focus. But that focus moves away from just providing the vaccination, actually, towards actually how we break the barriers that stop people wanting to partake in this program, especially in the communities that we need to get to. And I think Jeremy really stressed that, is that 20%, if we don't get there, we will see those horrible figures keep coming back. But even worse is if they've been left behind, the rest of the population may not be so sensitive to them. So actually, this is your time to really create that urgency that Rina's mentioned for the people that you really care about and see so much, to say, I want you to have this, and this is why.
And I think you're absolutely right. And I knew both of you would be great, because your passion just shines through, both of you, because I know you're having these amazing conversations with people. But actually, community pharmacy teams, one of the action points from watching this webinar is to go back and talk about this with your teams. I know one of my members of staff didn't want to have the vaccine. She waited till we'd all had it, and we didn't turn into chimpanzees, she said. She went and had it afterwards. So, there's hesitancy within our own teams and our own staff, which is really, really interesting to see. And it's so important to be proactive. So actually, identifying those people, like the carers who come into our pharmacies to collect medication on behalf of their elderly people they're looking after, just, start a conversation. Have you had your COVID vaccine? Are you considering having it? That's absolutely our role, to make sure these people, are getting that opportunity to have that informed discussion. And like I said, I was on a webinar with patients and members of the public, and actually what's stopping any of us as pharmacists running a webinar for our patients to say, have you got any questions about the COVID vaccination? Running a session like this for our own patients. We've all got very good on Zoom, and I think our patients have got very good on Zoom too. They'd probably quite enjoy that opportunity to ask us questions and to bounce ideas off people and listen to people's stories. I think that'd be a really, really good thing to do. I think the motto of the story is there's a lot of information, and I think Shirley and Jeremy did a great job of really creating a really digestible format of all that information for us to really have those absolute key points that we need to share with patients, so that we can go on to have that light bulb moment, as Bola said, and support patients to have that light bulb moment where suddenly they realize, what are my risks, what are my benefits, okay, I see where I am on this. And absolutely, as Ade said, being part of the COVID vaccination, the giving of vaccinations is actually a really lovely part of community pharmacy's role in this as well, isn't it? And supporting people, and they see, look, my pharmacist is giving me the vaccine, they're telling me to have it, they've had it themselves. There's no greater role model, is there really, than that for us. Just to end, I wanted to just do the poll again, if that's okay, because I'm hoping a lot of you who maybe weren't feeling so confident to answer those questions about religious aspects and the safety and the efficacy, and now are feeling a lot more confident. I know I am, and I shall be making my team watch the recording of this webinar because I think I've learned so much from it. So Simon, if you're okay to just share that poll, and while you're doing that, I just again wanted to say thank you very much to our esteemed guest today, Dr. Shirley Hopper. I'm really impressed at the presentation, and MHRA have worked at a great speed, haven't they, to really get this out to people as quickly as possible. So credit to you and your teams. I'm sure you've worked all hours, and I can imagine it's been very, very intense and stressful for you all, but thank you for your hard
Thank you, Rena, and thanks for inviting me.
And around it as well, thank you. I think your words really kind of hit home for me, definitely as well, about making sure we really target those main populations and those ones that are most at risk, to avoid this kind of continuing any longer. So the poll is ready if people want to start answering those questions. I'm going to definitely mark mine as feeling a lot more confident about all of these things, but obviously that's my answer. And we shall wait for the results. And Bola and Ade, thank you so much, both of you, for sharing your stories, and continue being absolutely inspiring and brilliant as you both are. I think this has hopefully spurred us all on even more to really get as many people vaccinated as possible. And we've all got a role and responsibility, haven't we, in ensuring that happens with our patients. Simon, have we got the results?
We have. I was just giving it a little bit longer, but I think we're plateaued. So I will share those results with you now. There you go.
So I'm seeing orange lines much higher up now, which is exactly what I wanted to see.
Definitely.
So that is really, really great. So hopefully we've hit the spot with this webinar for you all. Please do encourage people to the recording. Obviously, if there are any other topics that you'd like us to present on or create webinars, please do get in touch with us at events@npa.co.uk. If there's a few more questions on here that I haven't had a chance to answer, because I've just seen them creep in, I will do my best to get back to any of you in the next 5 days if there's questions that we haven't answered live today on the event. But again, I'd just like to say thank you all very much. Stay safe, stay sane, and keep being absolutely brilliant and promoting the message of vaccination urgency. Thank you all. Have a good evening.